environmental DNA and how does it help you to measure biodiversity?
It's a very good question. Yes, so hopefully, I can explain what eDNA is, and I can show you a quick video of it. So hopefully we can play that video now. Okay, so environmental DNA, what is it? So what I'm trying to do here, and this is me in a river just down the road last night, so I'm trying to go and collect some DNA shed by organisms into the water. And how do I do this? Well, first of all, I need to put on some clean gloves to make sure I'm not going to be capturing any of my own DNA in my sample. So that's the main thing. We want the biodiversity in the river, not me. So I put on my clean gloves. And then I need some sterile equipment, so nothing is contaminated with any other systems. So I have a clean syringe there, and then a clean Sterivex filter, a filter unit, which is what we'll use to collect the DNA. So as simple as those, that's all I need. And I get into the water body here. I want to enter the water body and sample upstream, so I'm not, again, contaminating my water with anything I might have brought in. And then I just, simple as that, I draw up the water using my syringe to an approximate 50 ml at a time. And then my filter unit can then be screwed onto that syringe, so it fits together nicely. And then as easy as that, I then push the water through my filter unit. So what I'm doing here is the DNA from that water is then sticking to that filter paper within that filter unit, and then the clean water is then coming out at the end of that filter unit. So I have my eDNA sample, and I think there's a closer up view, so you can see the little unit on the end. That's where my eDNA is being held on that filter paper, and that's what I'll extract the DNA from once I get it back to the lab. So it's as simple as that, really. I then need to prevent any further contamination that might happen to that sample. So I'll put on some red caps. You'll see that now. And I'll label the sample to make sure I know where it's from and who took it and when it was taken. So there's some downstream processes where I take this sample back to the lab, and I extract the DNA using some chemicals and some shaking to release that DNA and to clean it. And then essentially I have a tube of DNA from that water body. And with that, we can then assess it for lots of different things that might have shed their DNA into that water body. So it's a really exciting sample method, and I think this very short video, it's obviously not the full story, but it shows you how quickly you can go out and collect a sample. And with that, it means that our biodiversity monitoring can be kind of increased, like lots. Probably the most exciting part about having an eDNA sample is once you've done all the downstream lab work and you have your tiny Eppendorf tube of clear liquid or DNA, we can then assess it for lots of different things. And when we're thinking about biodiversity monitoring, previously we were always looking at things in isolation, perhaps fish or plants or insects, but now we can use DNA shed by pretty much anything that's in that river. We can try and look for it. So we can look for bacteria, we can look for changes in the bacteria communities in rivers, and we can also look for the fish in that same river. But also we can look for the mammals that maybe drank from that river. So rare mammals we might be able to pick up, and it's a kind of a unique game changer in how we might be monitoring our river systems, our aquatic systems, our terrestrial systems also. And in some cases, people also take footprint samples from snow and get the eDNA from that as well. It's a really exciting new monitoring method, and hopefully it will lead to a lot of new findings about our biodiversity and where they are, particularly hotspots for rare species and things that we might not see because they're so rare, but we'll be able to collect their DNA. So yeah, it's a really cool, exciting future for biodiversity monitoring.
Fantastic, thanks for that. It sounds fascinating. And I imagine it's if you're thinking about something like a tiger, or that's very difficult to spot in a real dense jungle habitat, and there's not many of them left. We know they're endangered species. Something like eDNA would be a really useful tool for that.
Definitely. I think these environments that maybe we can't survey as easily as a river in Switzerland, or these difficult, hard to reach places, there definitely eDNA can be a real plus, a bonus there, and hopefully it will lead to better understanding of where we have rare species, like how rare are they, and understanding systems better. It's really good. I really enjoy it.
And when you're talking about capturing that genetic information, what is it that the animals or the plants are leaving in the water or in the samples that you're able to detect?
So basically, it's like, I always, when I talk to people, it's like if you have a bath, right? If you get into a bath, you shed your skin cells, you shed sweat cells or anything like that. You essentially, it's not very nice, I know, but like think of that in terms of a river. Okay, a fish tend to be pretty slimy, so a fish will shed lots of slime, and other things will shed mucus, and you have different things being shed into the system. But essentially that's what we're doing. We're picking up any kind of evidence of these slothed scales or skin cells or anything like that from the species. So that's what we're collecting.
Sounds gross, but obviously really important information
yes,
to be collected.
I know. Yeah.
The glamorous life of a scientist.
Oh yes, definitely.
Fantastic. So